So, dear students, today our topic is factors affecting enzymes activity. So, already you know regarding the enzymes. So, enzymes are also known as the biological catalyst of the chemical reactions. So, without enzyme, so those reactions, so they require a lot of time and as well as lot of energy for catalysis of product. But in the presence of enzyme, so that is a required time is decreases as well as need of the expenditure of energy is also decreases. So that's why so enzymes are known as the biological catalyst. So regarding the enzyme catalyzed reactions, it will be represented as follow. That is a S. It indicates a substrate. So substrate can react with enzyme. So here the enzymes. So they will be having the various active sites. So these active sites it can occupy the substrate. Okay, so enzyme on their surfaces they will be having the active sites. So those active sites they are maybe in the natures of amino acids. Okay, they are made up of amino acids or any biomolecules. Okay, so those active sites so they can occupy the substrate. Then the complex will be formed that was known as the enzyme substrate complex. So after that, so where the catabolic reactions will be subjected or maybe anabolic reaction is subjected. Catabolic means to convert larger particle into smaller particles. Okay, so those are considered as the catabolic reactions, and anabolic means to utilize the smaller particle and synthesize the product or larger molecule. So those are known as the anabolic reactions. So here due to that is the composition of substrate with enzyme on the active sites, so maybe catabolic reactions occurs or maybe anabolic. Then after that, so enzyme was separated. Okay, and the product is living in that particular container, so where you can substrate can add up with the enzyme. So like that the finished product or the final product will be the forms. So here E represents the enzyme that can catalyzing the reaction, and S represents the substrate, and the P represents the product of the reactions. Okay, so here the example is, so we are adding maltose at a disaccharide with the enzyme maltase. Then so that maltase enzyme it can occupy the maltose in the active site that can subject it to the catabolism. So to catabolism of maltose disaccharide it can respond to formations of products like glucose. So then that glucose is required for our body for productions of the ATP. So this is the general enzyme catabolic reactions. So here the various factors, so they are affect on the enzyme activity. So from this figure, so you can easily understand so what is the factors commonly they can affect the reactions or biochemical reactions. Okay, कोई बोल सकते क्या? So anyone from this figure general, so these are the factors, sir, those can affect the enzyme reactions, maybe accelerate, maybe decrease. Okay, so you know, so number of enzymes, that is the concentrations of enzymes or velocity of enzymes. Okay, because enzyme is required for these reactions. If number of enzyme decreases means the product formation is also decreases. That means the rate of reactions decreases. Then as well as if enzyme concentration is high but still the substrate concentration is less, then also the formations of product is decreases. So these are the main two factors so that can affect the enzymatic reactions or biochemical reactions. Okay, one is the concentrations of enzyme and another one is the concentrations of availability of the substrate. If substrate less available means then there is no product, that much product can be formed due to lack of occupying the active site by a substrate. If enzyme is less means then substrate is more but enzyme is less, then also the formations of product or end product that is also decreased. So these are the main two factors, so they are mainly affect the enzyme activity. And as well as, so as you know, so enzyme so they are particularly active in the particular environment or conditions. Okay, if any condition is altered, for example, simply I tell you, so in our body, the small intestine is a site for digestions and metabolism and absorptions of the food. Okay, if any alteration in the temperature of the small intestine, then all the activities was decreases. Okay, if our stomach temperature increases or intestine temperature increases due to constipations or any other changes, then so digestion is not occurs, where you are suffering from the undigestion problem or indigestion problem, because so due to constipation, the heat was increases in the small intestine. So as the heat is increases, so where the enzyme activity was decreases, because so enzyme, so they are active in the particular pH that is around 30 to 40. If it is exceed more than 40, then all the enzyme active was decreases. Then as well as the pH of the environment, okay, that is also reflects on the enzyme activity. Particular enzyme in particular pH only, so they are active. If increases or decreases the pH, then that enzyme is inactive. So like that, so various factors, so they will be affect the enzyme activity. So now we can study one by one. Okay, so these are the various factors so that can affect the enzyme activity. First one is the, that is the concentrations of enzyme. Okay, so all the enzyme activity is depending upon the concentrations of the enzyme and concentrations of the substrate. If enzyme is more means, so more catabolic reactions occurs. If substrate concentration is more means, the more activity or final product will be forms. Next one is the temperature. So as the temperature increases, the enzyme activity is also increases up to certain optimal temperature. Then as well as there is water, pH, radiations, certain activators. Then as well as that is the end product is also affect the enzyme activity. Then also certain inhibitors, so they can affect the enzyme activity. Some factors are there, so they inhibit the enzyme activity, and some factors are there, so they can modulate or enhance the enzyme activity. Okay, first one is the concentrations of the enzyme. From the graph, you should be know, as the concentrations of enzyme increases, the enzyme velocity or the enzyme activity is also increases. It means, so the enzyme activity, okay, it is directly proportional to the enzyme concentrations. If enzyme concentration increases means the enzyme activity is also increases or the reaction is also increases. Okay, if enzyme concentration is decreases means the enzyme activity or the enzyme velocity is also decrease. It means, so the enzyme activity is mainly depending upon the enzyme concentrations, and this enzyme concentrations determination was helpful for the diagnosis of disease. Okay, in particular disease, so that enzyme activity was increases, so that increased enzyme activity is indications for the particular type of the disease. For example, that is the alkaline phosphatase. It is one of the enzymes. It is involved in the metabolism of the various products in the liver specially. Okay, in the case of inflammations of liver or any problem in the mineralizations of the bone, or the bone is suffering from the inflammations or injury, or even in the kidney. Okay, so where, so this enzyme activity was increases or the concentrations of the enzyme is increases, so that increased concentrations of enzyme was helpful for the diagnosis of particular type of the disease. Okay, even that is the, for example, certain hormones are there, is the bradykinin is there. Okay, then another one is that is the uh, atyl natriuretic peptide, that is ANP. Okay, it is also one type of enzyme. If its concentration is more in blood or in urine, that is it indicates some injury or problem in the heart. Okay, so like that, the way monitoring the enzyme concentration, that was helpful for the diagnosis of disease, because some reactions were increases due to that the ability of the enzyme or concentration enzyme is also increases. So due to that only, by monitoring the serum enzyme concentration, then only you can conclude or helpful for the diagnosis of disease. But the enzyme velocity Okay, so that our action that was directly proportional to the concentrations of the enzyme. So that's why all the biochemical reaction, our rate of biochemical reaction, so they are mainly depending upon the concentrations of the enzymes. The second one is the concentrations of substrate, that is the substance which subjected to the catabolic reactions by a enzyme, and such substance or molecules are known as the substrate, and the rate of reaction is also depending upon the substrate concentrations. As the substrate concentration is increases, the rate of reaction is also increases. It means the rate of reaction is directly proportional to the substrate concentrations, because so already you know, so the enzyme will be having the active sites. So this active sites is occupied by the substrate. Okay, or this interaction is occurs between the active sites of enzyme and substrate, then only the product is formed, then only the rate of reaction is increases. If the substrate concentration less means, then the enzymatic activity was present in the active site, that was also decreases. Lack of substrate, so there is no binding or complex between the enzyme and substrate. Due to that, the formations of product or the rate of reaction is also decreases. So that's why the rate of reaction is directly proportional to substrate concentration initially. So when the rate of reaction increases by addition, okay, initially the additions of substrate, so where the rate of reaction is increases by a first order reaction. So as the substrate concentration increases, the rate of reaction is also increases. Okay, that is by following the first order reaction. So later on, one steady is occurs or stable is occurs because of the active sites of the enzyme is completely occupied by the substrate. Okay, if that substrate is converted to product means next substrate can be occupied. Okay, number of enzyme site and substrate is equal on that state. Due to that, one state line is occurs up to certain concentrations of substrate. Okay, so where the rate of reactions is followed by the zero order reaction. So that's why the rate of reactions or the formations of products or biochemical reaction is also depending upon the concentrations of the substrate. So next one is the effect of temperature. So as the temperature increases, the enzyme activity is also increases up to certain temperatures. So here that is the temperature about 30 to 40 degree centigrade. Okay, so beyond 30, then also the enzyme activity is decreases. As you know from this graph, slightly the enzyme activity or the rate of reaction is increases as the temperature is increases. So maximum that should be obtained between 30 to 40. So after that increased temperature, the rate of reaction is decreases because due to denaturations of the enzyme. So after crossing the 50 degrees centigrade, the enzyme activity was decreases due to denature of the active sites, because the active sites on the enzymes, so they are made of the amino acids. So as the temperature increases, so those amino acids, so they are denature. So whatever enzymes are there, so they are all are denature when the temperature of the reaction is crosses the more than 50. So that's why the rate of reaction is mainly depending upon the particular type of temperature, that is around 30 to 40 degrees centigrade. So after that, the rate of reaction is decreases. So that's why always you maintain your body temperature about 30 to 40. If it is less than 30 means, then also the development of your body is retarded, then also you are suffering from the various kind of the disorder. If the body temperature is more than 40 means, then also we are suffering from the various kind of the metabolic disorder. So there is need to maintain our body temperature, that is the up to optimum 30 to 40 degrees centigrade were only. So all the enzymes, so those are involved in the biochemical reaction in the body or as well as the digestive reactions, so those are active only beyond the 30 to 40 degrees centigrade. If it is any increases, then all the process should be the abnormal. Okay, next one is the effect of pH. Okay, so pH is also influence on the rate of reactions, because so the particular enzyme activity, so they will be active in the particular kind of the pH. Okay, so below that pH, then also the enzyme activity was decreases, and above that pH, then also the enzyme activity was decreases. Okay, for example, that is the pepsin. So this pepsin is secreted from the gastric juice and present in the stomach, or secreted in the stomach from the gastric gland, and this pepsin was involved in the digestions of the proteins, or in the protein it was involved in the metabolism of peptide bond. Then it convert protein, larger peptide, into smaller proteins or amino acids. So that pepsin is active only when the pH is 1 to 2. Okay, so after eating a food, or food will be containing rich of proteins, and those protein, so they are enhance the acidity or acidic environment of the stomach, then the pH of the stomach becomes 1 to 2. Actually, the pH of the stomach is 4 to 5. Okay, in the normal conditions, when you are taken the food, okay, so after that, by lot of proteins in the food, so they are increase the pH, that becomes acidic, that is 1 to 2. So then only, so this pepsin enzyme is active. Okay, so then the pepsin is involved in the digestions of the protein. If the pH, if it is increases more than 2, then the pepsin activity was decreases. Or if the pH is less than 1, then also the pepsin activity was decreases, because pepsin, the maximum activity at optimum pH is the 2. Okay, so that's why unnecessarily you do not take the antacids or anti-ulcer drug, anti-secretory drug, because so they are enhance the pH of the stomach, so where the protein cannot be metabolized due to increase the pH, the pepsin actually was decreases. Okay, so due to that, we are suffering from the anemia or lack of hemoglobin, because hemoglobins are also proteinous substances. It is required the protein like a globulin, and that globulin is not supplied to your body due to lack of digestion of protein in the stomach by pepsin. So that's why we are suffering from the all kind of disorder related to the deficiency of the protein. So that's why the rate of reaction is, or the enzyme activity is also depending upon the particular type of the pH. So another example, so that was the amylase. So this amylase, it mainly secreted from the salivary gland, that was known as the salivary amylase, and also secreted from the pancreatic gland, so that was known as the pancreatic amylase. And these enzymes, so they are mainly involved in the digestions of the carbohydrates polysaccharide into monosaccharide, and that amylase is active uh, at the optimum pH, that is the 7. If the pH of the your oral cavity 7 means, then only that amylase was involved in the digestions of the carbohydrates, and also small intestine, if the pH is 7 means, then the amylase is active, then it is involved in the digestions of the polysaccharide into monosaccharide like a glucose, fructose, galactose. So that's why always you maintain the pH of your body, because the enzyme activity was depending upon the mainly the pH of our environment or body. So next one is the first few example regarding the list of enzymes and pH, that is the pepsin, it is source, that is the stomach, it is a protein in nature, so it optimum pH is 2. Then chymotrypsin, it is secreted from the pancreas, it is a protein, it is active at the pH 7.8. Then pepine, tropical, it is also tropical plant, that is involved in, it is a protein, so this pepine is involved in the digestions of the food, and it is active at the pH 7 to 8. Lipase, obtained from microorganism, substrate is the oil in nature or lipid in nature, and it is active pH is 5 to 8. Like alpha glucosidase, active in 6.6. Then beta amylase, that pH is 5.2. Then fructose franidase, that is 4.5. Then pectin lyase, that is 9 to 9.2. Like that, the lipoxygenase 192, that is active where the 9 and as well as 6. So like that different enzymes, so they required for the various biochemical reactions in our body, so they will be active on the particular type of the pH. Okay, next one is the effect of product concentrations. Okay, so the rate of reactions or enzyme activity that is inversely proportional to the product formations. Okay, so as the product formation increases, the enzyme activity was decreases, because if the product is, as you know from the reactions general, that is the enzyme reacts with the substrate, it can form the enzyme substrate complex. After that the product is releases, then enzyme is releases. Okay, so in our body in the particular site, if the product concentration increases means it can send information to the CNS, that is by a feedback mechanism. So automatically that CNS, it can decrease the activity of the enzyme. So as the product is decreases, then the CNS can enhance the enzyme activity. So that's why the enzyme activity is inversely proportional to the product. As well as in the chemical reactions, you can remove the product from the site of reactions. Otherwise, so those chemical reactions or biochemical reaction, the fermentation tank, so that was decreases, because as the product increases, automatically the enzyme activity was decreases, because it cannot be replaced by a from the active sites. If it is rapidly replaced from the active sites on the enzyme means, then only new substrate can be bind on the active site, then only so new product will be formed. So that's why the enzyme actually is inversely proportional to the concentrations of the product due to the active sites. If you replace the product means, then active site will be empty on the enzyme, then it can occupy the more substrate and involve the formations of the more products. Next one is the effect of activators. So certain enzymes, so they will be active in the presence of the certain factors. So those are known as the metal ion activators and as well as the coenzymes. So here the metal ions like magnesium, amines, then zinc, calcium, CO2, Cu, that is copper, sodium, potassium. So these are acts as the metal activator. So they can accelerate the enzyme activity. So due to that, the rate of reaction is increases. Okay, and certain presence of certain coenzyme like Fn, Fad, NADS. So these are the vitamins. So certain enzyme actually they are also depending upon the coenzyme. If the number of coenzyme is decreasing means, the enzyme activity decreases. If a coenzyme activity is increasing means, the enzyme activity is increasing. So like that it is similar to the metal ions. If the metal ions is available means, the biochemical reactions increases. If it decreases means, the biochemical reaction also decreases. For example, the blood coagulation process. So where various enzymes, around 12 enzymes, so those are involved in the regulations of the blood coagulation process in the body. So those all enzyme activity is mainly depending upon the calcium. So like that, the enzyme activity is mainly depending upon the activators, maybe vitamins, maybe metal ion. So on the basis of metal ion activators, so the enzymes, so they are mainly categorized into two types. One is the metal activated enzyme. So here the metal do not tightly hold by a enzyme. Easily it can transfer from one substrate to the another substrate or from one product to the another product. It cannot be hold occupy. But whereas metalloenzymes, so they can hold the enzyme tightly. Okay, okay, so they do not permit easily to exchange, and such enzymes are known as the metalloenzymes. And regarding the metal activated enzymes, easily, so those metal ions they can be transported from one molecule to the another molecule. So regarding the examples for metal activated enzymes, that is the ATPase, then as well as the analase. So ATPase require the metal likes magnesium, calcium as a cofactors, and then analase it is required the magnesium as a cofactors. So regarding the example for metalloenzymes, that is alcohol dehydrogenase, it required zinc, then phenol oxidase it required the copper, pyruvate oxidase it required the manganese, xanthine oxidase require the molybdenum, then cytochrome oxidase require the iron copper as a cofactor, and these enzyme activity is depending upon these particular type of the metal ions. If the metal ion decreases means, so these enzyme activity also decreases. If it is increases means, and these enzyme activity was also increases. So like that, certain activator or the enzyme activity is also depend upon the certain activators, maybe metal or maybe vitamins. So next one is the effect of time. So as the reaction time is increases, then the rate of reaction and formations of product is also increases. Okay, up to certain time, because as you know, so as the reaction increases, the temperature is also increases. Then after certain temperature, the proteins are denatured, because whatever the, okay, we can forms maximally, so enzymes, so all are protein natures. So after such a longer duration, so these protein will be denatured. So there is only one drawback, but remaining always. So as the time of reaction increases, the formations of the product is also increases. So that's why the time of the biochemical reaction is also reflects on the formations of products and as well as the enzyme activity. So next one is the effect of light and radiations. So as you know, so when we are exposed to the ultraviolet, beta, gamma, x-rays, so then only we are suffering from the various kind of the skin disorders or failure of the organs, because whatever the protective enzymes are there, so they are inactive when they are exposed to the ultraviolet, beta, gamma and x-ray. So that's why the enzyme activity is inversely proportional to the light and radiations. If they are more exposed to the light and radiation means, so their activity was decreases. If they are less exposure to the light and radiation means, so their activity will be the remain. For example, if the salivary amylase, if it is exposure to the UV rays, then the salivary amylase activity was decreases. So that's why you should always free from exposure to the ultraviolet, beta, then as well as gamma and X-ray. Otherwise, so enzyme activity decreases, then only we are suffering from the various kind of the metabolic disorders due to decrease the enzyme activity. Okay.